We're excited to kick off our panel on aging and longevity. Joining me on the panel is Robert Mansukhani, CEO of Deciduous Therapeutics.
So I'm going to speak today about aging and longevity. Our platform is around harnessing the immune system to treat age-related diseases. So said more simply, if we can understand the immune system, we can understand a number of different age-related comorbidities that happen, especially as we get into our older years. And more interestingly, if we can understand that process, we also have the ability as a biotech community to reharness the immune system to make it work for us again the way it was intended to when we were young. And so when you can do all that together, as the data will show you, I'll show you today, things can get actually quite interesting with how we can shift a paradigm for treating age-related diseases. So 4 parts to the talk today. First, I'm just going to give you a background on senescent cells. These are these pathological cells that we aim to remove, and I'm going to share with you, 1, what they are, and 2, their implication in age-related diseases. 2, I want to talk about how we can actually harness our own immune system to get rid of these nasty cells. 3 is the most important part, in my opinion, so that this is really where the rubber meets the road. So, in biotech, we often talk about interesting targets and mechanisms, but the question is, does it actually matter? And so I'm going to show you some disease models today where we've essentially reversed diseases in 2 very different ways using this approach. And not only do we reverse disease, when you think about the current paradigms and these indications, both metabolic and fibrotic disorders, it's quite a paradigm shift for how these things can be treated going forward. And then lastly, I'll talk about the path forward. So where are we now, and what do we do to get this thing into humans? So first, I want to define for you guys senescent cells. These are cells that have been irreversibly damaged. Cells becoming damaged is typically not a significant issue. Cells become damaged all the time. Sometimes they become repaired, but sometimes they become irreversibly damaged. In the case of senescence, what happens is these damaged cells, basically they stop replicating. So they go, said in more lay terms, what they're saying to the system is, look, something has happened to me that's bad. I'm not going to come back from this. I'm going to pull my emergency brake. I'm going to stop replicating. I'm going to exit cell cycle. I'm going to leave. Basically, I'm going to leave the party because whatever's happened to me is irreversibly bad. So, and the influences of bad can be a number of different things. It can be glucose. It can be reactive oxygen species. It can be telomere shortening. Could be acute injuries. There's a number of things that can cause these cells to become irreversibly damaged, but the effect is the same. Over time, they can become senescent. And when they become senescent and they don't get cleared by the immune system, they get very inflammatory. So they become very nasty. So basically what they're saying is, you should remove my immune system. The immune system does not respond in a case of disease or aging, and they stick around and then they become a problem. And so zooming on this a little bit more, so this is a snapshot of a senescent cell. It's in the chronic phase. Senescent cells at any given time in the chronic phase will give off about 60 different inflammatory cytokines. You may have seen on TV TNF alpha blockers, TGF beta blockers. That's about 2 of the 60 cytokines that are nasty and pathological being given off by senescent cells at any given time. But over time, as I said, the issue is not that they happen, the issue is that we can't get rid of them. And so what happens is you have this primary senescent cell here in the middle, and then it ends up making actually other healthy cells in the microenvironment also become damaged and senescent just by cell-to-cell contact. And this all culminates in driving chronic inflammation, fibrosis with tissue weakening, tissue degradation, and stem cell exhaustion. So the last one is particularly interesting because our regenerative capacity, as you know, is through stem cells, and those also become senescent when we can't get rid of these nasty cells. Now, these cells are implicated in areas of huge unmet need. First, these are indications, one that I could fit on a single slide, so there's actually more than just this, but I just want to give people a roadmap and snapshot of where this can be applied. These are all indications where senescent cell accumulation is thought to be causal in human disorders. These are also indications where senescent cell removal with preclinical studies by different methods has been shown to be impactful for treating disease. And as noted in the orange, these are also indications where massive market opportunities of high unmet need sit today. So these are the things, that roadmap of where you can apply senescence removal in humans going forward. Okay, so what are we doing about this? So first, I just want to give you the roadmap of the flow of how this works. So when you're young, your immune system is very active. You have, no senescent cells or very few senescent cells that keeps all the senescent cells at bay. But over time, with disease and with aging, the immune cells don't go away, but they become anergic. And what that means is they get slow, they get exhausted. They basically can't keep up with the demand of damage. And what happens is the balance starts to tip now. So now cells start to accumulate. The immune cells are not energetic. They can't really keep up. And so what we propose is with the small molecule immunotherapy, we can re-engage this immune process. We can remove senescent cells, and that will treat disease in a way that's revolutionary, as the data I will show you today suggests. So first, I just want to talk first 2 slides about how we identified this process, and I'll get the data right after that. So what we wanted to do is be very thorough in our platform approach. So we wanted to look at different senescent cells from very different tissues. So pulmonary tissue of lung disease, adipose tissue from fat tissue from metabolic disease, and lastly, diabetic pancreatic tissue from autoimmune disease. And we took senescent cells out of them. We isolated those cells. And we did something called single cell RNA sequencing, which basically allows us to listen and analyze these cells at a very close level. We actually can look at their gene expression. We actually can look at what are they trying to tell us that's unique towards the immune system. And so since it's a short talk today, I'm not going to spend time getting into all the nuts and bolts of how we did this, but fast forward about 9 months with our academic partner, what we figured out is that a very specific, very often overlooked type of immune cell called the invariant natural killer T cell is responsible for the removal of senescent cells. This is the publication we put out last year. If you want to read it, this is the title right here. You can Google it. It should be open access for you guys to look at. So by the way, also one of the coolest names that immunotherapy is possible, invariant natural killer T cells, kind of tells you already what they're responsible for. Okay. So now the premise around the company and then the data. So the hypothesis then became, okay, let's activate these NKT cells, let's re-engage them. That can remove senescent cells. And then if we do that, this NKT-mediated senolysis, meaning senescence removal, will represent a paradigm shift for treating multiple age-related disease classes. Okay, so let's get to the data. The first data set, I'm going to first explain the model, and then I'll take you through the data itself and what we showed. So these are mice, essentially we put them on a Western diet. So at week 6, we put them on this high-fat diet, high-fructose diet. And for 4 months, we just put them on the diet. So we're not treating the animals with the therapy. We're just letting disease set in for a period of 4 months, a very chronic time period in a mouse. At the end of that 4 months, we give them 1 dose of this NKT activator drug. So we're activating the immune system in these animals where it's been suppressed. We then look at target engagements. We want to know that we've actually hit the immune target. We then look at, does it remove senescent cells? And most importantly, does it actually treat disease? So from the first dose is over here, week 22, and within 10 days, we see an improvement in disease. So here's the data. So the first one, we look at just target engagement. So did we actually hit the target with the molecule that we wanted to? So these are activation profiles of natural killer T cells. The standard here is a normal mouse, no high-fat diet, just normal weight, normal mouse. The second mouse here, the high-fat diet vehicle, is the one that has been given the diet but no treatment. So it's the untreated animal from the disease. And lastly, the one that we've treated. So we see a really nice induction now, the activation of these NKT cells. Now we know they're active, they've been mobilized, they've expanded, they're ready to go. Also on the same day, we see that we've now reduced senescent cell levels back to age-matched control levels. So these mice that are still twice the weight, metabolically compromised, have senescence, used to have senescence everywhere, now have a senescence burden that's been reduced all the way back to normal mice. You see the animals that we have not treated have a high senescence burdens in the adipose tissue. Then we look at insulin resistance. So the mice that we've treated now become less insulin resistant. The goal is always to get back towards the red, the more standard animal that hasn't had any disease. And then what that culminates in also is an improvement in glucose tolerance. So one of the main drivers or one of the main characteristics of metabolic disease is, glucose intolerance. So we want to see if we challenge the mice with a bolus of glucose, a high dose of glucose, can we make them metabolically normal once again? And so you see these animals in the orange that we've treated, despite still being twice the weight of a normal mouse, despite still having, high-fat diet-induced disease, start to become metabolically normal again within just about 10 days from a single dose. And so the paradigm shift here is we've given this dose once. So it's a single treatment. The effect lasts, you see, this is day 10 right here. This effect actually lasts for a number of weeks after this. So what we envision in the future is that instead of taking diabetes or metabolic, condition pills multiple times a day, you would basically have a cleansing of your senescent cells every quarter, and that would more effectively treat disease while also reducing the pill burden quite significantly. So that's the paradigm shift. Number two, we wanted to look at a second model, very different than metabolic disease. We look at lung fibrosis. This is a standard model for idiopathic pulmonary fibrosis. So in this model, what happens, we take mice, we give them a bleomycin injury to the lungs. So basically what that does is it causes fibrosis and senescence and restricts lung function in these mice. And then again, we see at day 10, after induction of disease, can we dose the drug? 4 days after that, can we expand the NKT cells and mobilize them to get rid of these senescent cells that we've put into the animal? And then 3, does it actually improve fibrosis? So this is lung fibrosis. So one of the primary endpoints in a mouse is, can you actually halt the fibrotic process in these mice? So again, left to right, we see nice target engagement of the NKT cells in the lung, especially versus controls. We now see that the senescent cell volume in the treated animals versus the untreated animals is now significantly reduced in the lung tissue. The fibrosis score has improved dramatically. So the mice that even after, in these models, by the way, we induce disease. We don't treat it early. We don't treat it prophylactically. We actually treat well after disease induction. So we see a really nice reduction of the fibrosis of these animals, even after 10 days of having given the disease. And then even in a separate academic study, what we learned is that even at a lethal dose of bleomycin, most of the animals that we treat, even with a single dose, way after the induction, will survive that study. Okay, so what are the advantages and why should you care? First, actually, let me just highlight one more point. So again, same paradigm shift here. IPF right now is a really nasty disease. It's poorly managed, poorly treated. You take about 3 to 6 pills a day. They do minimal effects on fibrosis, nasty side effects. This, again, has been given one time. We expect this effect to last for several weeks, if not more, at a time. So we envision about a quarterly treatment to treat lung fibrosis going forward for this disease. Okay, so what's the value prop here? So one, we have durable endpoints. So you have intermittent dosing, so you can take a lot fewer pills, and based on the data I've shown you today, they should be a lot more effective. 2 is this is a systemic approach. So you think about age-related comorbidities. You often have more than one disease happening at once. This is a systemic way to ablate senescent cells all throughout the body. 3, this is the innate mechanism. So if you think about, traditional drug delivery, you want to think about the way nature intended for things to happen, the way for nature intended for things to be treated. And so when you do the hard work to figure out what that is, it tends to be a much more effective, but also much more safer approach. And so we really like that we're just piggybacking on nature to do this. 4, precedent for safety. So NKT agonists have been tried before in the clinic for very specific oncology studies. What was shown there is that there were no significant adverse events from the mechanism of activating NKT cells. It's robust, meaning that it didn't seem to matter in the data sets how we induce disease. One was through a 4-month diet process, one was through a single injury to the lung, but the effect of these drugs was still the same and still quite significant, but it did not matter how the disease actually came to be. And then lastly, attractive cost of goods. So there's small molecule immunotherapies. These are 2 microgram doses we've given it to mice. That projects to be around 3.5 milligrams in a human given once every couple months. So you're talking about 4 to 5 treatments per year at a very small dose. So small molecules, are inherently much less expensive than most cell therapies, obviously, or antibodies. So we have a really nice cost of goods here that can play for us well in the future. And what it means for the global communities is this is a treatment for everybody, not just for the haves or the have-nots. This is something that can affect everybody. Okay, so in summary, we therapeutically harness natural killer T cells to ablate senescent cells and treat disease. This in vivo data supports a significant paradigm shift versus traditional drug development. Future milestones include platform expansion and first-in-human studies. Thanks to a couple of our pharma sponsors for our non-dilutive lab sponsorship, Eisai and AbbVie, 2 of the biggest pharma groups in the world. Here's some of the recent press we've had over the past year. And thank you again to Scott and Astor Perkins for having me on. If you want to learn about partnership opportunities, if you're a scientist looking for your next job, reach out about job opportunities, or if you're interested in investment inquiries on our next round, please send me an email or add me on LinkedIn. Happy to chat with about any of those items. Thank you.
Great. Thank you very much, Robin. I appreciate that. Now, as someone that has to take, certain pharmaceutical drugs on a regular basis for prediabetes, for instance, I mean, the thought of intermittent dosing periodically to activate these NKTs to blue-ray senescent cells. I mean, it's mind-boggling. It really is. It's kind of a holy grail. I guess the question that comes up, it's kind of twofold, really, is how does this adversely affect pharmaceuticals that really monetize off patients taking medication on a daily basis, right?
Yeah, it's a good question. Yeah.
But at the same time, they're also the ones that's interested in your studies, interested in the potential benefits of these NKTs.
Yeah, it's a good question. And if I could just zoom out on your questions, basically, there's probably more money to be made if you're taking something more consistently that's more expensive, right? And so we're coming along saying, look, this is going to be less expensive. You also don't have to take it as frequently. So I understand, get the question. I would say, our job as biotech entrepreneurs, as a company, is to do what's best for patients. It's not about, the monetization will come after we show that the impact on patients is significant. We paid attention on our indication selection process to understand where the payer tolerance actually exists for these types of drugs also. So I think of it kind of in both the commercial lens, but I first and foremost think about it in the patient lens. It's not exactly for me to worry about if other people are upset by something that's better and less expensive. I think that as an entrepreneur, that's always what you're shooting for. So that's how I think about it. That's how we'll consider this going forward.
Okay. And, one thing you mentioned is that there are cell-specific senescent cells, right, in the beginning of your presentation. But one of the things that you're starting to show through a couple of these studies is that these NKTs are effective across a wide range of diseases. And I wonder if you could explain that.
Yeah, so what happens in senescent cells accumulate through a number of different induction methods. There's a lot of things that can cause a cell to become irreversibly damaged. So you think like a huge cell, there's 10 plus things that can cause a innocent cell to go, okay, I'm irreversibly damaged because something has happened to me that's really bad. And so those cells end up exiting cell cycle, and typically in a healthy state, they end up getting removed by the immune system. The issue is that when the immune function fails, those cells kind of go from this filter of different induction methods of senescence all the way to a very single, very consistent profile. And this DNA damage-induced senescence profile. And that DNA damage-induced senescence profile is what the NKTs picked up. So it doesn't matter to the NKTs how the senescent cells got there. What matters is that they all kind of filter into this unique signature that the NKT cells can recognize. And so part of our rationale in showing the platform is that we can induce this in different ways. It didn't matter, but the effect is still very strong. And the reason the effect is very strong is two reasons. One is because the NKT cells are very effective at getting rid of them. Two is because the senescent cells are very nasty, right? They're pathological in significant ways. They're inflammatory, they drive fibrosis, they exhaust stem cells, but they also have this paracrine effect where they make cells in the microenvironment also take on their bad poison and become senescent and become irreversibly damaged. So it's like when you just clean out all the bad actors in the tissue or in the organ, you're laying the groundwork for things to come back to normal. You're basically resetting back to homeostasis, and stem cells can again take root and do their job. It's kind of like spring, after a long winter. That's how we think about it.
And I think the area that you're working on is not only fascinating, but in many ways it's actually profound, because especially as it relates to the effect to stem cell research and development, it's pretty significant, right? So somebody who's, let's say, in their late 70s or 80s, with the proper obliviation of senescent cells, they could, in theory, have healthy stem cells for reproductive new organs, for example, or new limbs, whatever it may be, right?
Yeah, and it basically allows your function to return. But it gives your body an opportunity to do what it wants to do. It kind of gets all the bad stuff out of the way. So yeah, it's and this will be somewhat heterogeneous, indication. It's not going to reverse all diseases. It's going to maybe in some cases have a halting effect. In some cases, caught early enough will have a reversal effect. There's going to be, with patient populations with comorbidities, you're not going to solve every disease here, obviously, but you're going to be able to at least mute a significant amount of age-related comorbidities. That's the goal.
Mm-hmm. Now, some of the studies that's been done on mice are very promising, to say the least. With that said, there's also been research that's come out about things that's been published even in things like Nature that all of us kind of trust, is that oftentimes a lot of these smaller animal testing is really not necessarily a good or perfect indication of how it's gonna behave in human in vivo. So talk to us about what we can anticipate as you go from mice to maybe larger animals and eventually to humans.
Yeah, this is a very good question. This is the age-old question in biotech, right? So is the animal model that we are showing that this thing works in, can we trust that it will show the same thing in a human being? This is what we think about, every day. So first, I just want to segment and say, not every animal model is created the same. Some models are thought to be very predictive. Some models are thought to just be models. So we just have to take it all kind of together. Part of the rationale in showing the diseases the way I did in the way we did the first two POCs is to show that the diversity in the induction of senescence didn't have an effect on how successful we could be. So if it was a injury through a chemical bleomycin, it still worked. And that, by the way, that is a gold standard model for IPF, believe it or not, is, injuring a mice with bleomycin and giving them fibrosis in senescence in the lungs. So we always focus on gold standard models. But to your question, the way you will really know is in a human study, does it work or does it not? And if you have quantitative endpoints, it won't be up to anybody's subjectivity as to whether or not something is working. In the case of the high-fat diet data I showed, that one mirrors, human disease quite well. I mean, that's the high-fat diet. It's high fructose, high fat. That is a large part of Western culture, and this is what happens to people in these cultures. I don't think I've had in this deck, but there's a deck we show in one of our presentations where we show the linking of senescent cell accumulation in human tissue to disease. And so the indications that we're focused on all have a significant presence of senescent cells on the leading edge of disease in the human condition. And so we focus on those areas. We use the models in those areas to tell us if this thing's going to work or not. It's also studying the paper that I alluded to, that where we actually had done a study early on where we took human blood, isolated NKT cells, activated them in a dish, and put them against human senescent cells, and showed that the NKT cells in a dish from human blood, so a human in vitro study, would selectively and successfully clear human senescent cells. So we've de-risked this a little bit just by looking at basically a human tissue sample in addition. So this shown the same phenomenon works quite well.
Mm-hmm. So I know I believe in past conversations that we've had offline is that your team has been working on identifying the right molecules that's going to have really the best results. And I believe you guys are transitioning to allocating those R&D efforts to the specific molecules that's going to be the best candidates. So as you guys get ready for that safety clearance, talk to us about how a lot of these molecules are actually fairly well known and already been pretty well vetted from an FDA perspective, and how that kind of de-risk, I suppose, the process moving forward.
Yeah, that's a good question. So, as I alluded to in one of the last slides, the idea that an NKT agonist, an NKT activating molecule, there have been a few of them already in clinical trials already. So those are different molecules than what we have. We've made subtle tweaks and improvements in formulation advancements and how we dose, et cetera, to get these molecules to work for our purpose. But the point in sharing that information was that the mechanism, meaning that the mechanism-based safety profile looks pretty clean. So the idea that you can go in and activate NKT cells in previous clinical studies has been shown before. And what was interesting about those is that there were no significant adverse events reported in those clinical studies. So basically, when people have their NKTs activated already, we already know that there's not going to be a significant adverse or great tox effect or safety effect from this mechanism being woken up in your body. And the reason for this is a couple-fold. So one is the molecules that we've designed are highly specific. So what I mean by this is, it's too quick for a talk like this, but there's a receptor that's unique on the NKT cell. We model our molecules to bind specifically to that receptor. The receptor being unique, what I mean by that is it doesn't exist in any other cell type. And so if you can understand that receptor on the NKT cell, you can get really specific, nice activation of these cells. So that's one. And then two, when we activate these cells, it's typically quite transient. So we're not talking about, these blasting on your immune cells for weeks or months at a time. What we see actually is that we see a peak of activation around day 4, but by day 7, there's essentially no activation. So it's kind of like a nudge, like, hey, go do your job. You're already there, go do your job. But once you're done with your job, you can go back to bed. And that's how they activate and then come back down to resting states.
Mm-hmm. Fascinating. Let's talk about the value or the additional aspects of IP, which is around the data. So as you guys conduct additional studies, joint studies, partnerships, and apply to different diseases, there's a tremendous amount of data that's being developed. Talk to us about why is that valuable and how can that be leveraged for other purposes?
Yeah, so additional data we're working on. So one is, the IP in the company sits with the compositions and the methods. So novel, unique NKT agonists to clear senescent cells to treat a whole variety of diseases. The additional data we'll generate will be around more indications. I didn't talk more about those today. Those are still, under wraps in the company, but we showed metabolic disease today. We showed pulmonary fibrosis. There are a number of other indications, as I mentioned on that roadmap, where this approach can be impactful. And again, we want to keep pushing the limits on different ways to induce senescence, different types of disease classes, but typically all things that are more or less associated with age-related diseases. It's interesting to think about, muscle deficiency as we age. It's interesting about heart performance, cardiovascular performance, kidney function, et cetera. So those are all areas that we're now thinking about other ways to apply this. And we've got a couple more programs under wraps that I can't share in a live forum, but certainly brain aging and dementias are also quite interesting to us.
Okay, great. Talk to us about where you guys are as a startup, both in terms of funding, but also maybe additional partnerships and path to licensing.
Yeah, so we are, so today we started the company about 2019. It raised about 16 million to date. We'll do another round sometime next year, which will achieve funding for the first human study. Partnerships, we've had some early discussions. As I mentioned, we've picked up some non-dilutive partnership funding from both AbbVie and Eisai. We've got a couple of the conversations going that I probably cannot share in this forum, but there's been a lot of pharma interest. Some things that we've liked, but haven't decided to move forward on quite yet because I think it's early for us. So the idea is always to from a company perspective, I think my job is to ensure we continue to generate excellent, compelling, paradigm shifting data. And when you have that, you can think about partnerships for certain indications, or you think about a partnership that's more global that encompasses the entire program. So there's some conversations we've already had with a couple groups here and there. So, like I said, in the meantime, it's really about us advancing our molecules and to generate data that's really compelling. I think if we do that, the rest of it will naturally flow.
Amazing. So I want to thank Robin for a terrific conversation on senescent cells. Please give him a round of applause. We now transition to our climate change and sustainability track to talk about our panel on sustainable cultivated seafood.